Welcome to Short Talk with a Scientist. I'm Dr. Teja Klančič, your host, passionate about nutrition, metabolism, and the fascinating world of gut microbes. It is my pleasure to introduce Dr. Marie-Claire Arrieta, a remarkable scientist, a mother of two, and a passionate advocate for science communication. Through her research, she has been uncovering the ways in which changes in the gut bacterial and fungal composition impact immune diseases such as asthma. Beyond research, Dr. Arrieta is dedicated to sharing scientific knowledge, writing books for the public, and participating in documentary projects. Claire, it's such a pleasure to be here today. I can't believe it's been 7-8 years since I was in your office and you asked me for my candidacy about early life microbiota development and how it's linked to the immune system. And that really sparked my interest. And then I went on to read your book, Let Them Eat Dirt. And that book is really fascinating, like describing how children in a too sterile environment are predisposed to certain diseases. So I'm really glad you took the time today to share your stories with us, to share your scientific discoveries. So thank you so much.
Teja, it's wonderful to see you again. It's wonderful to see that you've embraced science communication. As you know, this is something close and dear to my heart as well. And it's just great to talk to you about science and what we've been up to. Well, you know that we continue to be really interested in the gut microbiome, this complex community of microbes that we have everywhere, but mainly in our guts. And specifically, we're really interested in what happens very, very early in life. We're born without any microbes, right? But then the second that we're born, we start acquiring them. And we actually know not a lot about that initial process, who comes first, what happens after, and how that initial establishment of these pioneer microbes has a lot of say in who's going to come after, and importantly, how our different systems are going to develop. And you alluded to this too, that microbiome is really important in terms of the way our immune system develops, but also the way our metabolism develops. And you remember that from your PhD, the way also our brain develops. So our microbes are definitely more than just bystanders that we get to share a body with. They're also more than the agents of diseases. And there's a lot that we need to learn about what they do early in life. And that's what we've been doing. So one area that we become really, really intrigued is what happens with the microbiome of babies that are born prematurely.
Okay.
So normally, you know, gestation, it's about 9 months, but babies can be born at 5 months of gestation. And you know, prematurity continues to be this great burden globally all over the world. There's a lot of babies that are born before they're supposed to. And depending on how early they are born, that can carry a long-term health burden as well. So these babies are at a higher risk of developing immune diseases, respiratory diseases, also neurodevelopment impairment as well. And knowing what we know now about the microbiome and how it can contribute to the normal development and the normal physiology of all systems, we really posed the question, well, what about babies that are premature? They carry this significantly higher risk. What about their microbes? So we have started to look at their microbiomes, and so have other people around the world. First thing, their microbiomes are very different. They almost don't look like a baby's microbiome at all. For a number of weeks, their microbiome is very, very altered. And eventually, after a few months, it starts becoming a bit more baby-like, or what we would see in babies that are born healthy and that are born at term. And we started a study, is now quite a large study here in Calgary, called the Alberta BLOOM study.
Okay.
And we recruit these babies soon after they're born at all stages of prematurity. So whether they were born in the 5th, 6th, or 7th, or even the 8th month of gestation. And we've been collecting samples to understand how their microbiome develops. We've started answering some questions related to the ecology of this ecosystem. Not only which are the microbes that, set camp in there first, but also which are the important ones, and which are the ones that may be beneficial, right? Which are the ones that you actually don't want there. And we've been also involved in a clinical trial of the use of probiotics in this population of babies. Sometimes they get probiotics, sometimes they don't. And we found through this study that if they do, and if they get actually a probiotic with the right type of bacteria,
Exactly.
that can really shape their microbiome in a good way. And it can set it into a trajectory closer to what the health, term-born would be.
One question, before giving them probiotics, do you actually check whether that particular strain is already present in them, and then, or do you do some kind of screening prior to giving them probiotics?
Yeah, we do, because they rarely, we start looking at them and their microbiome soon after they're born, and it takes a few days before they can start taking probiotics. So we have a good understanding of the same baby before and after probiotics, and also a comparison of babies that don't get probiotics versus babies that do. Unlike the probiotics that can be given to the adult population or even to older children, when given soon after birth, these strains, depending on which ones, with some probiotic strains, can actually stay there. And remain there because they got there first. And this is something that we're finding with premature infants. And by doing so, we're finding that there's certain strains within the microbiome, certain beneficial strains, that really stabilize the community. Not only do they stay there, but they can also stabilize the diversity of the community, and also from a functional point of view, the substance that the microbiome is producing when they're given probiotics, they're also beneficial in terms of their immune development and whatnot. So super interested in what happens to the microbiome of premature infants. The nice thing about the study is that it's a very long study. It's a 5-year study, so we're gonna continue following these babies. Our older babies are now 4 years old, so some of them are about to even start preschool. And by bringing them back every year, we can then look at how their immune system is developing, and how their neurodevelopment is going, how their learning is going, how their motor skills are going. And we're super thankful to the parents that are so dedicated into bringing their kids back to our study so we can learn more about it. So that's one side of what we've been doing. The other side, you alluded to it a bit, has to do with the microbes that are not bacteria. We're really interested in asking, well, who else is there? Because we know that in any other environment, whether it's in aquatic environments or in forests, you name it, microbiomes are composed of all types of microorganisms, not just bacteria.
Ecosystem.
That's right. And we're really interested in fungi first, because we know that fungi are everywhere in the environment, more so in certain parts of the world than in others. But also we know from studying the type of infections that sometimes some of them can cause, that our immune system has a way to react to them that is different than the way they react to bacteria.
Oh, wow.
And they actually react also pretty strongly to fungi. So it seems like we have evolved ways to perceive fungi in a different way. So if we now know that the microbiome is critical in immune development, but so far we've only studied bacteria, we thought, well, if we know that our immune system reacts differently to fungi, maybe we should pay attention to them and see if they are helping to develop other areas of our immune system early in life. So we've been doing this both in humans also, and also in in mice as well to understand a little bit more, right?
Right.
And what we have found first in humans is just like with bacteria, we have certain species of fungi that come first, and then that changes depending on different factors. For example, if the baby is given breast milk or baby is given formula, nutrition, just like with bacteria, is very, very important, right? As soon as the baby starts eating complementary foods or solid foods, then the microbiome or the fungal microbiome starts changing as well. We've been really interested in understanding the interactions between fungi and bacteria. And as expected, or I've seen in other ecosystems, they interact. Some of them will benefit each other. Some of them actually will antagonize each other, right? So we're understanding a little bit more about what happens in the early life microbiome by also considering other microorganisms like fungi. We now understand also that one of the factors that really can alter the microbiome early in life are antibiotics.
Same like bacteria.
Yeah. Okay. And the thing with antibiotics is that they are, bacteria killers. They will kill bacteria. Well, because we have a bit of a microbial jungle there. Fungi actually take advantage of that, and they will overgrow. They will take over for a bit until the antibiotic is gone, right? And what we have found was that the longer a baby is under antibiotics, the stronger this fungal overgrowth gets. And this is perceived by the immune system as we expected in a different way. And we're now using the power of being able to use animal models. We're understanding that yeah, indeed, certain fungal species that overgrow during antibiotic treatment of babies, they can trigger, they can wake up certain aspects of our immune system that are necessary for diseases like allergies or asthma to develop. So meaning that fungi are contributing to that unbalance that needs to happen early in life for certain bodies to develop allergies, like food allergies or like asthma or any of those. So very intrigued with what fungi are doing. Another story related to fungi that we found shouldn't have surprised us, I guess, because it had been shown for bacteria, but it had surprised us. And it is the fact that fungi can also change our metabolism. So how much we put on weight, or mice put on weight. So first we found this in babies in one of those longitudinal studies that follow babies as they grow, and we published that indeed there is a fungal signature, if you may, that is linked to overweight in children. And this is early in life, even before they develop obesity or risk of obesity. And then when we then ask the question where we then take some of these fungi that we find in humans and we put them into mice, and we find that indeed the fungi can really change the way animals put on weight or don't put on weight on a regular diet or on a high-fat diet, for example. So we're really intrigued by how fungi may be doing that.
That's so interesting. And when you were doing fecal transplant, because the mechanisms you're describing, it's very common to see with the bacteria, right? So the fungi seem to do the exact same thing. So I'm really wondering, when you do a fecal transplant, do you transplant a mix of bacteria and fungi, or do you just separate the fungi? Because
that'd be a good question. How do you tease that apart? Right. And this was part of our design. So luckily here at the University of Calgary, we're very fortunate to work with a facility of mice where you can grow mice under very defined conditions. We know exactly which are the microbes that different mice have. Yeah. So instead of doing a complex transfer of, dozens or hundreds of microbes, we have animals that only have bacteria and we know which ones they are. And then we have animals that have the same bacteria, but with one or two species of fungi. And then that allows us to compare the difference that having a fungal species makes. We can also then treat those animals with antibiotics and then simulate an overgrowth of fungi and see what happens in animals that receive the antibiotic but only had bacteria, or the animals that also had a fungal species or two, but were given antibiotics. So that's our approach to tease that apart.
That's fantastic. Claire, thank you so much. This was so insightful. I've learned so much, and I cannot wait to share this with a wider audience.
Great.
Thanks a lot.
Thanks.